electron transport chain is made of 3 globs, or we call complexes more. It's better to say the word complex than globs. We have, and this is the one that I want you to, the slide that I want you to study from for the way the electrons are flowing. Remember that each species in each complex is a electron holder, and you can see that whenever it's in the reduced form, it has the electrons, and whenever it's in the oxidized form, it's given it up. So here, NADH has the electrons. It's passing it on to an enzyme-bound coenzyme called FMN. Then it passes line. The NAD no longer has it, and then the FMN has it. So it's a bucket brigade. It's really the best way to think of it. So we're passing electrons from FADH2 and NADH all the way to coenzyme Q. Coenzyme Q travels within the membrane to complex 3, gives it to more iron sulfur centers, another cytochrome C, which has iron cofactor, to a different cytochrome C, which actually goes out of the membrane back into it and into complex 4, which goes to a cytochrome A, A3, and a copper ion. I'm not really going to ask what is the specific order. Does it go to the iron sulfur center or the FMN first? I'm not going to ask about that. I just want you to know what's in each complex. So you don't have to know iron sulfur, then cytochrome C1. You know, I just know that those are in complex 3.
How do you read that again? So I get the first 2.
So here, to read this, when it has either the hydrogen or in the reduced form, it has the electrons. So here, this has the hydrogen, it's reduced, it's passing it on to FMN. Now, this is a reaction. You can think of the arrow going down. NAD no longer has it, FMN has it. FMN H2 has the electrons, it's going to react with FS, an iron sulfur center. That's in the oxidized form. It's going to give it back up. Now the iron sulfur center has the electrons. The FMN no longer does. It's ready to accept more. Just like when you're in a bucket brigade, you have the water, you pass it off. And then you're going back. When you go back, you no longer have the water, right?
So the electrons would go from the iron sulfur to the...
So now in the reduced form, it has the electrons.
Right.
It's passing it along to coenzyme Q. Now see, now it doesn't have it, and now this does. So when they're touching here, that's where the passing of the electrons happens. You're pouring the electrons from this guy into this guy. And so on, down the tube. This is what they really look like. It'd be kind of funny if they were colored like this. But they're globs along our inner membrane. And you can see these phospholipids, these little balls with the little lines here, those are the phospholipids that make up our membranes. We're going to study that later on this semester. But here's complex 1, complex 2, complex 3, and complex 4. Notice that complex 1, 3, and 4 span the membrane, but complex 2 does not. Now, we said in the previous slide that complex 1 and 2 supply electrons to coenzyme Q. UQ is another way to write coenzyme Q. So coenzyme Q is also known as ubiquinone. UQ and coenzyme Q are the same thing. So notice that I said before that complex 1 and complex 2 give their electrons to coenzyme Q or ubiquinone, and it travels through inside the membrane. And then to complex 3 gives the electrons to a cytochrome C. This cytochrome C can go into the intermembrane space and travels back to complex 4, which then gives its electrons to oxygen. We're going to get back to the differences between them, but what do you think? All right, so these are the membrane's made of phospholipids. The little ball is a phosphate, and the waggly tails are hydrocarbon chains. what do you think ubiquinone or coenzyme Q looks like, or its nature is, if it can go through this part? Yeah, it's hydrophobic, or it has a part that makes it mostly hydrophobic. So you guys are learning. See, if you knew that, then you've got a huge concept from this semester that I'm very proud of. All right, but cytochrome C can go in and out, so it has both hydrophobic and hydrophilic parts, so it can go back out. All right, so what we're going to do now is take a look at each complex, and I want to mention that the order that I have on the slides, the cartoons that I have on the slides, aren't necessarily going to reflect exactly what I have on slide 15. So the order might be different. There might be some extra things because I'm trying to teach this in a sort of basic level in this class, although the only things I could find were a little bit more complex. So study from slide 15 and know that these other ones might be a little bit more complex. So here in complex 1, there's some new information I want to tell you. Okay, so we know that we're going to take the reduced coenzymes from the citric acid cycle in glycolysis. We're going to take that reduced coenzyme. We're going to pass the electrons to our enzyme-bound FMN. We're going to take a look at FMN in the next slide. But then it's going to pass it on to these orange and pink things, or yellow and pink things called iron sulfur centers. We're going to take a look at those too. So we're passing the electrons and passing. So there's a bunch of iron sulfur centers. There's 6 to 8 of them in here. So I want you to know NADH, FMN, iron sulfur centers, then to coenzyme Q. In that, though, in the passing of the electrons from the NADH to FMN to the different iron sulfur centers, you are going to take 4 hydrogen ions and pass it from the matrix into the intermembrane space. That's very important. Guys, you okay? Okay. So you're going to take four protons from the matrix and pass them into the intermembrane space. It's very, very important that we know that there's the amount of free energy that is released by passing those electrons along gives enough off so that we can pump protons from the matrix into the intermembrane space. So if you want in your notes, write matrix, intermembrane space. That might be a good thing to do. All right, so we have our coenzyme Q. It's picked up the hydrogens, the hydrides. So you can see those hydrogens are holding those electrons. All right, let's take a look at one of the new electron carriers. This is FMN, and it picks up the 2 hydrogens here and here. I'm not going to ask you the structure. I'm just showing you. Just know that FMN is in that one. But I just wanted to show you. It picks up the 2 hydrogens there. Does anybody need?
Yeah, can you see that again?
So I'm not going to ask you to draw the structure or even to recognize it. Just know that FMN is another coenzyme, a new coenzyme, that can pick up and carry hydrides, electrons. And it's going to pick them up here and here, right here and right here. That's where they're picking them up. But I'm not going to ask you that. I just want you to know that it's a coenzyme that is carrying electrons. Iron sulfur centers. So cool. All right, so these are non-heme iron proteins. So because so many, when we encounter iron in our enzymes, in our proteins, so often when there's iron in those proteins, it's in a heme structure. We'll look at that again later, like as in hemoglobin. It's in that heme structure. So we need to point out when iron is present that it's not in the heme structure. So that's why we call it a non-heme iron structure. So they're different. You can have a 2 iron or a 4 iron. There's 2 different forms. And they're held together or held to the proteins through sulfur bonds, sulfur coordinate covalent bonds. But what really is doing the work is the iron. So when it picks up electrons, it's going from iron 3 to iron 2. When it's giving it off, it's going from iron 2 to iron 3. All right, so do I need to say that again? When the ions of the iron sulfur centers are picking up electrons, it goes from the oxidation of +3 to +2. When it's giving them off, it's going from +2 to +3. So it's being oxidized. So reducing, picking up the electrons, going from 3 to 2, oxidation 2 to 3. You definitely need to know, I'm for sure 100% going to be asking that question. So the work that's being done in the iron sulfur centers is the iron. What makes it have a slightly different standard reduction potential is going to be the environment in that non-heme iron protein, in that iron sulfur center. The amino acids that are surrounding it will make those irons have a different standard reduction potential, different affinity for electrons.
Can you say that part one more time?
Sure. So what's doing the work of the iron sulfur centers is the iron. All right, it's being oxidized and reduced back and forth. But whenever you see iron, it's doing that. What's making this iron different from other irons is the chemical environment. And that chemical environment is the sulfurs around it and the amino acids around it. That would be a good question. Why would the irons that you see in the electron transport chain have different standard reduction potentials? That would be a great question, wouldn't it? That would be, yeah, I like that. I like that question. Okay. The sulfurs around it, the amino acids, the chemical environment, really. If you say chemical environment, I'd give you the credit. I'd love you to say the sulfurs and the amino acids. But really what makes the irons have a different standard reduction potential is the chemical environment. Is it kind of freaky? Look at that. It's kind of weird. All right. We didn't talk about Coenzyme Q very much. How many people go to GNC and buy CoQ10 so that they can get more energy out of their workouts? Huh?
My dad does.
Yeah, be careful. He has to take CoQ10. Does anybody know why it's 10, not 8 or 6? What does a 10 refer to? Does anybody know? Anybody that's taken any other biology classes, you might have heard this. You didn't hear about this in any other biology class? You did, but you don't remember? Okay. The 10 is this isoprenoid unit. Do you see how it has the brackets around it and then this N? That means that it repeats. And for mammals, it repeats 10 times. Bacteria repeats 6. So be sure that you don't go to GNC and buy CoQ6. It won't work for you. I'm trying to be funny. It's really not working on a Monday morning. Okay, so this is CoQ10 because the isoprenoid unit repeats 10 times. Didn't mean to say that again? It's CoQ10 because the isoprenoid unit repeats 10 times. So if this repeats 10 times, that's wicked long, right? So remember, the CoQ10 is swimming in that, along the membrane, in that hydrophobic part. So this part is what's making it hydrophobic. This ring, the quinone form, it's not so hydrophobic. It's got 4 oxygens on it, right? So it's not really hydrophobic, but that isoprenoid unit makes it, excuse me, hydrophobic. So here what it's doing is it's resonance stabilized to accept 1 electron on its at a time, but then it can ultimately accept 2 electrons as hydrides and makes our coenzyme Q10 or ubiquinol or reduced form or hydroquinone form. In this class, let's keep it simple. I'm going to call it CoQ, reduced form of CoQ. All right? But you might, I have license to use UQ. You might read it in other textbooks as UQ. I'll try to stick to CoQ on the exam. But I think it is important to see that it is called different things. All right, you guys ready? We know everything about complex 1 now. NADH, we've seen that structure before. FMN, we've now seen that structure. Carries electrons. The iron sulfur centers have the iron that's doing the work. And now it's passed along to coenzyme Q. Complex 2. Some classes don't even cover this, all right? Because it does not have enough free energy in passing the electrons from the FADH2 to iron sulfur centers and then to coenzyme Q. Don't worry about this heme one, all right? Just know FADH2, iron sulfur centers, coenzyme Q. It does not have enough to drive ATP production. That means there's no protons being pumped from the matrix to the intermembrane space. So some classes don't even cover it. Now, in this one, we're showing that it does span the membrane. I tend to believe that other picture that it's not spanning the membrane. There's another source of FADH2, and that is going to be through fatty acid oxidation that we're going to see later on. So the first complex had enough free energy. This one is the only complex that doesn't provide enough free energy to pump protons. This is the only one that you cannot pump protons. Complex 3. This one is not a great picture because it's too busy. Complex 3, there's more to complex 3 than what we're going to talk about. The electrons are going to be taken up by our, let's just look at this part right here. All right. This is the Q cycle. We're not going to look at this part. So you can X out this part of this picture, but you want to take a look at this part. So you're going to take the electrons from the reduced form of coenzyme Q. You're going to give it to iron sulfur centers and cytochrome C1. Then that's going to pass it along to cytochrome C. Sure, you're going to take it from the coenzyme Q, this QH2.
That's the same thing as coenzyme Q.
Yeah, QH2 is the same thing as coenzyme Q. And then it's going to pass it to iron sulfur centers in cytochrome C1. Then it's going to pass it, that cytochrome C1 is going to pass it along to cytochrome C. We're going to talk about the cytochromes in 1 second. But this does provide enough free energy to drive the pumping of protons into the intermembrane space. So 4 protons are pumped. Does provide enough free energy to pump. Okay, so let's talk about this Q cycle. I'm not going to talk about, I do mention it here, but I'm not going to test you on it or talk about it at all on the exams. The cytochromes are a group of proteins. They do contain the heme prosthetic group, but they're not meant to carry oxygen. All right? It's the same prosthetic group, but what the iron in the prosthetic is doing is being oxidized and reduced instead of carrying oxygen. That's kind of cool. It's doing double duty, that heme prosthetic group. So here's the heme prosthetic group. Remember, the iron is in a coordinate covalent bond. Do you guys remember what I said about the coordinate covalent bond, where you're looking at metal ions that have empty d orbitals, and both electrons come from lone pairs from, say, nitrogens. So see these nitrogens right here? Those are resting in empty d orbitals of our iron. So it's holding it in. So here, you can see that you have this ring structure holding this iron in. So this is basically a heme group. Now, you have these numbered positions, and you can see in these different positions, we have different groups. I'm not going to ask you the difference, all right? I'm not going to test you on that. But just know that the way we get the cytochrome C1 and the cytochrome A to have different standard reduction potentials is to change what groups are on these positions. Bless you. So we're passing along from cytochrome C1 to cytochrome C to cytochrome A3 to A. You know, there's a lot of passing along. They all contain this heme group. But what's different is that between the A cytochromes and the C cytochromes is that they have these different groups on there in these different positions. So that chemical environment is making it a little, this iron have a little different standard reduction potential. So look at it. It's like, okay, here's the first layer around it, the nitrogen. And then the second layer around it's still the same. It's this third layer around the iron that becomes different, and only slightly different. So that third layer around the iron is enough of a difference to cause the standard reduction potential of the iron to be different in these different cytochromes. That's a profound effect. If you guys haven't studied NMR, you might not have the appreciation for that. But those of you that have studied NMR and know the effect on, say, hydrogens or carbons of chemical environment that's even kind of far away from those carbons and hydrogens, that it may be new to you. So I think it's kind of cool that there's this real application of the chemical environment. All right, so that's the cytochromes. Complex 4, again, this slide is busier than I want it to be, but it's going to take this cytochrome C that we just reduced, and it's going to be oxidized. It's going to give it to different cytochrome A's and to copper ions. That's all I need you to know. There's cytochrome A's, and then we just talked about the difference between cytochrome C's and cytochrome A's, but cytochrome and copper ions. Remember, copper ions have the oxidation states of 1 and 2 instead of 2 and 3. So 1 and 2 for copper instead of 2 and 3 for iron. So again, cytochrome C is going to give it to copper ions and cytochrome A's, 2 different cytochrome A's. And then what is the ultimate electron acceptor? Oxygen. So we're going to give it along to oxygen to make water. So complex 4 is the link to oxygen. If you don't have oxygen around, can't complete this. We do have enough energy to pump 2 protons from the matrix to the inner membrane space. So 4, 4, and 2. And complex 2 is not doing really anything. In fact, complex 2 is so boring that some slides I said don't even show it. So here we have our NADH, FMN, iron sulfur centers, to coenzyme Q. Know that complex 2 supplies more coenzyme Q reduced form. That's really what you want to know about coenzyme Q. The complex 2. And so here then we're going to take the coenzyme Q, and don't worry about the Q cycle. Go to iron sulfur centers, cytochrome C1. Cytochrome C can come down to give cytochrome A. And this one doesn't have the copper in it, so maybe you might want to put your copper ions in there. Here's the free energy. You can see, I'm not going to focus on this. I'm going to focus on this one. This is complex 1. This is complex 3, and this is complex 4. So you can see that we get a lot of free energy in these 3 complexes. Complex 2 is not really on here. Yeah, because it doesn't take the FADH. So it's not, complex 2 is not addressed here because we're not taking it from that FADH. But you can see that the free energy from these 3 complexes is significant. And that free energy is going to be used to pump those protons uphill. And so we're going to see next what we can do with those protons.